Hi, I'm Jennifer from OpenHelix. Welcome to this tip of the week. If you're viewing this movie somewhere other than at our blog, be sure to check out the post at blog.openhelix.com to see the full discussion with links and references. In today's tip, I want to feature an Allen Institute for Brain Science tool that has been recently updated. The tool's name is the Brain Explorer, and I'm going to access that by clicking on this Human Brain tab. The Brain Explorer is a really amazing tool because it allows users to compare expression in two different human brains using a 3D viewer, as we'll see in a minute. So once the human brain page loads, I can either select data by using this tag cloud. What I'm going to do for this example is enter a gene name, and I can do other searches other than gene, and click search. Sometimes searches at Allen Institute tools take a little while to load. That's because they are drawing from such a huge granular amount of data. And now that our CDK1 data has loaded, here's what we see. There are some empty areas which will be filled after we select a data point to view. There are some tool and function access points on the right, and then there are our results. As I said, we need to initially load some data points to work with. I'm going to click here on our map. And what we see is information about that data point. So here's a structure hierarchy showing us where the expression occurs. There's gene information, and there's information about the brain in which this data comes from. If we click on this donor link, we'll see a list of all the different donor brains that we might get data from. We want to look at the Brain Explorer in today's tip, so I'm going to click on its logo. The Brain Explorer software must be downloaded to your computer before you can use it. If I had not previously downloaded it, this message would lead me through that process. Since I have downloaded it, I get this security message, which I'm going to click on Allow. Now, what we see is the Brain Explorer. On the left is the 3D graphic view of two different human brains showing the expression of the gene we're interested in. On the right is a structural hierarchy. The color coding here corresponds to the color coding along the heat map. In this area we have a gene list. We can click on either one of any of these lines to go back to displaying that expression data in this area. You can have multiple genes. You can also have multiple data points for the same gene. The viewer currently is showing the two different brains, O1 and O2 brains. If I want to focus, let's say, just on this O1, I can click on this magnifying glass here. And then I get a much closer up view of this brain. If I want to go back to the double view, I click the X. If I want to switch from inner structure, which is shown by default, to outer structures, I can do that in the Atlas menu. I can rotate this brain to get a better view of what I want. The highlight tool allows me to click on an area of the brain. In the graphic, that area will be highlighted in green, and in the hierarchy it will be highlighted in gray. In this tip, I've only had time to touch on a few of the Brain Explorer functions. I hope I've excited you enough for you to go to the tool and investigate even more of the functions. But it really is a cool tool to be able to explore expression in two different human brains. And this, again, is from the Allen Institute. Thanks for joining me today.